Debt Nation, where we are busy putting death out of business. And great show. My favorite politician, Ben ZION2020 here, and Logan. I'll let Logan introduce himself in a second. Really, really great mind. We're really, really proud and happy to have Logan here. Thank you very much, Logan, for coming onto Debt Nation to talk about brain emulation, full brain emulation. But before we do that, let's thank our sponsors for this episode. That's Thrivous. That's Lincoln Cannon. He's doing a fantastic job there with geoprotectors and nootropics. And you know the deal. The link is in the rules are in the description for a great prize. If you want to win some nootropics, that's $5 all the way to $160. The more people that share the video, the more nootropics you're going to get. So if you want to enter that competition and get some free nootropics, you know what to do. Go and follow the link, rules in the description, share it, say thanks Thrivous, thanks for all that nootropics, fantastic. And I am Transhuman David Kelly, also sponsoring this episode. Thank you so much, sir. You're doing a great job. Now, let's rock into the episode. So this is about full brain emulation. You know, when you think about that, that's a very big topic, you know, especially when you're considering humans, considering the complexity of the brain. But first, baby steps, we're gonna start with full brain emulation of fruit flies, and we're going to explain why that is. So let's cross over to Logan to open up a barrage of information. Firstly, Logan, introduce yourself, to your education, you know, how you just graduated. Fantastic, well done to hear that, going on to further education at some other location. Just explain who you are. Welcome to the show, sir. Thank you so much for coming on. It is a real honor and a privilege to have you here. You know, the pre-interview discussion between these two gentlemen here has just been absolutely fascinating. There's lots and lots, and we'll go into some of that stuff as well during this interview, this discussion, this show. But Logan, please enlighten the audience on who, what, when, where, why, how, and then we'll talk about fruit flies. Thank you so much, sir.
All right, thank you very much. So my name is Logan, and as you know, and I started doing scientific research when I was in high school in synthetic biology, and that eventually evolved into a nice publication in ACS Biochemistry. But sort of partway through undergrad, I became very interested in neuroscience, and particularly in the idea of whole brain emulation. And so I made a transition to the field of connectomics, which is basically developing new tools to map the brain. Specifically, I'm interested in the connectomics, which it has the resolution sufficient to image individual synapses and dendritic and axonal fibers of neurons, so subcellular resolution. And I am currently doing research on some X-ray microscopy-based approaches to connectomics, and also developing a way to combine that with expansion microscopy. Furthermore, I have some additional ideas which I may work on sometime in the future around developing a way of a new approach to connectomics, which may involve sparse imaging that is sort of repeated. But I won't really go into that a whole lot right now because that's just sort of the inkling of an idea. Anyway, so my primary areas of expertise are synthetic biology and nanotechnology and connectomics, and so I apply a lot of that in my research. And I'm specifically interested in whole brain emulation because I sort of see it as this way of both exploring our humanity and potentially ensuring the survival of our species beyond the near-term future. And so I'm very excited to contribute to the field of connectomics, since it's so integral to understanding how our minds work and informing whole brain emulations. So with regards to what Steele mentioned about insects, I recently published a paper on that was sort of an editorial journal article in the journal Biological Cybernetics that was sort of a proposal for developing whole brain emulation of insects, and it focused specifically on Drosophila melanogaster, which is the fruit fly, and the honeybee, Apis melliferae. And so this is the reason that I proposed this is because a lot of the existing work in whole brain emulation has been sort of premature and has garnered some backlash. There was a project called the Human Brain Project, which is still going on, but has sort of shifted direction from being mainly focused on whole brain emulation to being more focused on sort of a diverse array of neuroscience innovations. And that project, Henry Markram, who I'm a big fan of, but a lot of people have sort of had some backlash against some of his proposals because he actually proposed human brain emulation in 2010 and said that it would happen within 10 years. And of course, it hasn't happened within 10 years. And so I'm trying to propose this idea of insect brain emulation as an intermediate step, which can demonstrate to the larger scientific community and the public that doing simulation of complex biological systems in an integrative fashion is possible and can yield great benefits, while still being something that is feasible in the near term. And I made a very rough estimate of within 20 years, but I like to say that it is possible that it could happen within 20 years. It's not definitely going to happen within 20 years, since it really largely depends on the allocation of resources and how much, and to some degree luck, but also how we organize our institutions and our research around the goals of integrative biological simulations. And so I encourage people watching to check out this paper. It's in the journal Biological Cybernetics, and it's under my name. Yeah, so I'd like to discuss some of that here on the show, and perhaps adjacent topics as well.
Yeah, absolutely, and I'm really looking forward to getting to the meat and potatoes of this. The mapping of the structural neural anatomy with you, and I really like this idea, this intermediate step. Sometimes scientists do this. They in space they want to build the Dyson sphere straight away. They don't want to do the intermediate steps, or they want to build the AGI before the narrow AI. I mean, they try and jump a little bit because they get excited, and I like this idea of bringing it right back to something like the fruit fly, and then working forward from there. As you get more, as you work on more complex animals and complex creatures along the way, shellfish or something, clams or whatever. So yeah, fantastic. And you mentioned restructuring, social norms and institutions and stuff like that. That's where Ben ZION comes in. Ben ZION, you haven't always been a politician though. You've worked very closely in biological fields and laboratories at schools and such. What's your interest in your Ben ZION 2020? What's your interest in full brain emulation, connectomics? And this sort of neuroscience idea. Why are you here on this panel, Ben ZION? And it's lovely to see you, Ben. Thanks for coming on, and good luck in Chicago. We're counting down 1 day, 13 hours. Safe travels up there, and you're gonna destroy that field. So we're looking forward to that as well. But it's good to see you, Ben.
Well, thank you, sir. Yes. So in the pre-interview, we had a fascinating discussion about the Openworm project. And this highly autonomous entity, which has a mind, and what are the implications for computer science in having access to a thing like this in silico? And that is really the question in any of this. I like the idea of whole brain emulation, but I have a brain. You guys have brains. There's many billions of brains that could be being put to better use, arguably, today. Maybe all of them. We should look at the effect on as much on using these things to improve systems more generally, and not focus on one particular outcome, but rather focus on any kind of takeoff effects or improvements that could derive from these sorts of things. And the breakthrough in the Openworm project is, as Logan mentioned, it's a lot fewer neurons being emulated there. I wonder. I have a question for Logan. There's a few of these kind of left field questions already, and in the pre-interview. But what about a fetal brain emulation? What about starting at the earliest phase of a human and trying to emulate something like that? The operative notion being that we want to have something, a brain that can grow and integrate into super intelligent systems and make advanced algorithms truly autonomous and robust.
That's a very interesting question, Ben, and thank you. So with regards to sort of a early developmental stages, I think that is a very interesting idea and something that hasn't really been explored hugely in the field. I think that there could be a lot of benefits that would come out of that, especially given a way of a way of sort of bridging that to the types of computations that may go on as the brain develops further. And specifically, I think that one of the great benefits of looking at the developmental perspective is that, I mean, it's sort of a way to build a brain. And so and I think as neuroscientists who are not developmental biologists, I mean, there are some neuroscientists who specifically study neural development, but I think when you're a neuroscientist who is more focused on adult brains, you end up oftentimes neglecting the power of seeing how the system developed in the first place. And so understanding using by using early structures, you may be able to gain a better understanding of later structures through simulating the developmental processes. And so I think maybe it wouldn't necessarily be the cognition in the early fetal brain that would necessarily inform a great deal of artificial intelligence, but I think that understanding the architecture of the fetal brain as well as the molecular rules which lead to the wiring of that brain as it develops could yield great results and could greatly accelerate the process of understanding how our minds work, in the sense of we could see how the early brain is organized, and then we could do sort of systematic studies of gene expression, receptors, gradients, targeting of axons, how different glial cells influence the development of brain tissue, and from there be able to derive general wiring rules which could allow us to understand at a coarser level how the brain ends up being built in the first place. And using this, we could definitely potentially accelerate the progress towards being able to develop very human-like emulations, and from there we could use those emulations to better understand how our cognitive systems work and inform artificial intelligence, as well as inform a lot of other fields.
Mm-hmm.
Well, yeah, I mean, isn't the point of the fruit fly because we okay, so it's not quite reverse brain engineering in that way, because you're I mean, with this fetal brain idea, but isn't the point of the fruit fly to make this an incremental system of research, as opposed to trying to do what Ben says and put the cart before the horse? Is it too big a step to do that, to do fetal research before you've done the fruit fly, for example?
I think that it is. It might be too big of a step if your fetal research is explicitly stating its goal as human whole brain emulation, or deriving deriving enough rules to reconstruct a human brain without the explicit wiring diagram, because that's essentially the approach that the Human Brain Project has taken. They're trying to use wiring rules and sort of the general organizational principles of the brain to reconstruct something that does not have the exact connectivity of any existing brain, but is rather follows the same sort of almost follows the same developmental principles. And so I think that kind of research can be very valuable, but I hesitate to say that would be to use that research as the central vehicle for whole brain emulation, since it could since like you said, it could be seen as putting the cart before the horse and has sort of a negative effect on the economic landscape of this kind of computational neuroscience work. But like I said, if you were able to do that, I still think that it could yield a lot of valuable insights. And it could also sort of cross over with some of the insect brain research, in that, especially if you were doing developmental insect studies, so that you could better understand sort of the molecular context of how insect brains are organized. You may be able to derive insights that would be very helpful for biomedicine and for the design of neuromorphic architectures, as well as for perhaps even weak artificial intelligence, prior to really explicitly telling the world about these vast and grandiose goals of whole brain emulation.
Yeah, I understand that. Before we move on to the ability to do that and changing the institutions to do that, and also the morals and ethics behind let's talk about imaging technology. So Logan, open up on imaging technology. What does imaging technology have to do with this incremental process?
Right. So with imaging, I mean, imaging is sort of the central way by which we understand the structural aspects of the brain. And I sort of ascribe to a view of neuroscience as, if you're able to collect all the parts and know how the parts work, you can then integrate them together and test them against the biological reality to see if they work the same way in your model, and then iteratively improve the model by comparing it to the biological reality, making adjustments, and then cycling back to look at the biological reality again, doing the comparison, making more adjustments, and so on and so forth. And so with imaging allows us to establish the wiring diagram of the brain. It allows us to see where the synapses are connected, even to some degree how strong the synapses are, as well as the morphological properties of the neurons, which are very important for their computations. You see, the one thing that's oftentimes overlooked in artificial intelligence is the vast computational power of even a single neuron, because the dendritic arbor, or the dendritic tree, has a great deal of computational complexity. There's basically what happens is that there's a depolarization when the neurotransmitter binds to receptors on the ligand-gated receptors on the postsynaptic terminal, and then there's this depolarization of the membrane because the ion channels open, and then ions flow through the membrane and are and cause a change in potential. That's electrical difference across the membrane, and then this potential difference can actually diffuse along the membrane by affecting other ion channels, which are sort of spaced along the dendritic tree. And there's a variety of different mechanisms by which this happens. There's what are called calcium spikes and NMDA spikes. I won't go into all the details there, but sort of to give you an intuitive picture, a neuron will receive a synaptic input, and then there will be sort of this propagation of voltage across the dendritic tree, which decays as it goes along, but can also be enhanced by the molecular localization of various types of ion channels, as well as the inputs from more neurons. And so there's this very complicated process which happens in the dendritic tree, and there's actually been a variety of computational studies which show that it can implement some fairly complicated operations within a single neuron. And so the morphology of the neuron is very important, because if you know how the dendritic tree is structured, you're able to construct what are called multi-compartmental Hodgkin-Huxley models. And multi-compartmental Hodgkin-Huxley models are a type of mathematical model which has been shown to be quite good at predicting the diffusion of voltage across the dendritic tree and the eventual summation that leads to the firing of the neuron. And I shouldn't say summation here because it's a very nonlinear process. It involves partial differential equations coupled with some very complicated ordinary differential equations, but it does have a long history of being quite good at predicting neuronal activity. And although it's not perfect, it can be an excellent way of emulating neurons in silico. And so there are ways of however, okay, so where was I? I was talking about sort of the imaging of the dendritic trees. We want to see sort of several different components. We want to see the structures of the individual neurons in the brain, and we want to see how they're connected and where they are connected on each neuron. So, like if you have a neuron that has a bunch of different dendrites, you'll want to see where on the dendrites the different excitatory, inhibitory, modulatory, etc. inputs are connected, because where they are specifically linked can have influences on the computations. You also want to see the sizes of the dendritic boutons, or sorry, not dendritic, the presynaptic and postsynaptic boutons, because that can be used to predict the strength of the synapses and sort of the weights that need to be applied to them. And so a lot of these imaging technologies are very, very important because it allows you to see what's going on in that sense. However, there is sort of a piece missing when you talk about this stuff, which is the gene expression. Because as I mentioned earlier, I see this as sort of assembling different pieces together and knowing how the individual pieces work and trying to reproduce emergent properties by iterative improvements. The gene expression is missing in the purely imaging picture unless you have specific markers, either fluorescent or nanoparticles, or something else, which is able to tell you what types of genes are being expressed in the neuron. Because, as I mentioned earlier, the ion channels are very important in particular, as well as other receptors which can bind neuromodulators. And so in order to really understand how neurons work, and in order to parameterize the multi-compartmental Hodgkin-Huxley models, you need to know the types of ion channels, you need to know the types of neuromodulator receptors, as well as probably some other gene expression properties. And so there are some emerging imaging technologies which can actually tell you this stuff simultaneously. And I can go into, if you'd like, I can go into sort of an overview of the different imaging technologies and their strengths and detriments. But I think
let's talk about that. So let's talk about the hardware. So where is the best hardware to sort of to image the dendritic tree and these neurons and stuff? Is it in the United States, and what is the imaging hardware, and where are we at, and how are we going to what companies and what are we working on to get better hardware, and where do you see the hardware in the future, etc.?
Yeah, so that's a great question. And the sort of the leading connectomics technology right now is electron microscopy. A beam of electrons is scanned over a sample, and either the absorption of the sample or the scattering of the electrons allows us to reconstruct an image. However, this has to operate in 2D, and because of this, electron microscopy ends up being very slow. So essentially what happens is there have been a lot of automated electron microscopy technologies that have been developed where there's automated processes of either slicing or ion beam milling, which will essentially chop up the tissue sample from whatever brain of interest or neural system of interest you're looking at into thousands of ultra thin slices, usually around on the order of 25 to 40 nanometers if you're using a knife-based slicing technique, or on the order of 10 nanometers if you're using an ion beam milling technique, which is essentially you're just shaving off the top layer after you image it using a beam of ions, and then you image the next layer, then you shave off that layer, then you image the next layer, you shave off that layer, etc., etc. Whereas with the knife-based serial section electron microscopy, it's more like you actually use an ultra sharp knife, oftentimes a diamond knife, to shave off very thin sections of the tissue, and then you can actually, in some cases, preserve these sections and go back to them later in case you need to correct defects. But anyway, with regards to your question about how, like, what institutions are available for this kind of thing, there are a variety of sort of large centers that have that have some pretty extensive electron microscopy hardware. So one of these is the Allen Institute. They have a set of, I believe, 5 transmission electron microscopes, which are able to operate continuously without outside interference 24/7. And there's an automated slicing system, and sort of an automated sample collection system, which allows these electron microscopes to take large tissue samples and automatically image them at very high resolution in a way that's much faster than we would have been able to do even 10 years ago. And so this is, however, this still can only reach one of the big milestones that was recently attained was imaging an entire 1 millimeter cube, which is still pretty small in the grand scheme of things, but is sort of a major achievement in the electron microscopy field. The thing about electron microscopy is it's very high resolution, which is part of in addition to the slicing, is part of the reason that it takes so long. But it does give you very good images. However, there's also another weakness to electron microscopy beyond it being slow, which is the fact that there's not a whole lot of technology out there for attaining molecular information from electron microscopy. That is to say, the gene expression, the ion channels, things like that I mentioned earlier. And so, in order to get that, there have been some efforts at correlative electron microscopy and color electron microscopy, but they haven't yet been integrated into the larger workflow and the automated systems, and there may be some challenges there. But I am optimistic, especially given that electron microscopy has developed so rapidly over the past 10 years. It's gone from it taking a decade, or it hypothetically being something that would take a decade to image a 1 millimeter cubed volume of tissue, to it taking 6 months over the course of 10 years, which is a pretty big jump. And I imagine that there is likely going to be continued changes which lead to ever greater volumes being able to be imaged. And there is some talk even now of doing whole mouse brains. And I think that there might also be some new innovations, perhaps even something that I would be interested in working on with regards to color electron microscopy, or some kind of correlative system where you take an image of using fluorescence microscopy, and then you take the image with the electron microscopy that could reveal the molecular information necessary to parameterize the multi-compartmental models that I mentioned.
What's the most common species or that they're currently using electron microscopy on? Has it been fruit flies? I mean, you say they're looking at mice, right? So what have they been slicing at the moment?
So primarily, there's sort of two different areas. So there are mice. Primarily, people have been looking at mouse cortex. So the outer surface layers of the brain, which are thought to be responsible for a lot of the higher functions that mammals are capable of carrying out. And so people have been looking at mouse cortex. However, the fruit fly brain has actually been, there's actually an entire electron microscopy volume already available for the fruit fly brain. The fruit fly brain is pretty small. It's less than a millimeter cubed. It's significantly less than a millimeter cubed, actually. It's about, I think, like 160 micrometers by 300 micrometers by for something or other. Anyway, it's not important, but it's fairly small, and it was actually in 2018 there was a full electron microscopy volume available, and by now the computational image processing technologies have advanced to the point where we've segmented about a third of it. And by segmentation, I mean we have filled in where the cells are and determined and been able to separate out which cell is which, as well as determine where the synapses are. There was a recent collaboration between Google and Genelia. Genelia is a large institute which focuses on neuroscience research as well as some other biological research, and in this collaboration, they were able to create a connectome of 25,000 neurons in the Drosophila brain electron microscopy volume, and they're planning on finishing that, which is the full fly brain has about 100,000 neurons. Some estimates are slightly higher, but usually people say 100,000 neurons.
So I've got
so I guess it's about a quarter.
I've got two more questions on imaging. Oh, sorry, but you could finish your point.
I was just saying basically that it looks like the fruit fly brain, there's going to be a full connectome available fairly soon. However, there's still going to be some information we need to create a simulation of a fruit fly, which is more along the lines of those ion channels and neuromodulator receptors that I was talking about.
It sounds like you know, like almost any advance or any technology, your first one is your highest cost and your most time invested, and after you get the fruit fly down, the next one will be cheaper, quicker, faster. There'll be you know, but I got two questions. So one, before you jump in, Ben, but I'll let you jump in a sec. But one. Okay, this is an emerging technology question. So with imaging, specifically on imaging, what do you think will be the influence of, say, nanotechnology in the imaging space? So that's my first question. Emerging technologies, you know, going inside, different imaging. And two, why image at all? Why take tissue samples and all that? Why not let a computer sort of generate what it thinks is the brain structure? You know, because we've got these algorithms now. For example, you feed it inputs, you ask for an answer, it generates the answer. We don't know how it gets to the answer, but it might get to the answer. Do you think that we might be able to just skip taking tissue samples at all, and maybe a computer will pop out answers that we need, not necessarily having the information? So we might be able to skip the actual learning process and let an AI or something like that generate answers and then work back from the answers. So emerging technologies and then skipping the entire process of science using algorithms. Emerging techs first.
Right. So emerging technologies. So this is actually an area that I'm very, very interested in, because personally I'm not as set on electron microscopy as some people are. And I actually have been exploring a variety of alternative approaches. Specifically, my research is currently focusing on trying to combine expansion microscopy with X-ray microscopy. And what this means is, so I'll tell you what these two technologies are. Expansion microscopy is a method of infusing tissue with a swellable hydrogel, which physically enlarges it, thus increasing the effective resolution. And X-ray microscopy is a non-destructive imaging modality, which is essentially equivalent to a CT scan. So it's a 3D non-destructive. You don't have to slice the tissue. It creates a 3D reconstruction just by rotating the sample and shooting X-rays through it. And the thing about X-ray microscopy is that it is very fast. Laboratory X-ray microscopes have speeds that are they're similar to light sheet microscopy, but synchrotron-based X-ray microscopy is sort of a emerging area in neuroscience where you can actually use a type of particle accelerator called a synchrotron and you can take advantage of the very brilliant beams of the synchrotron to image things very rapidly. However, the resolution of this is on the order of 500 nanometers rather than on the order of, say, 20 nanometers, which is closer to what electron microscopy can do. And so I'm currently investigating ways of potentially expanding tissue, making it physically larger, and then sort of filling in the gaps with nanoparticles in order to facilitate rapid synchrotron-based imaging, which could actually image an entire expanded shrew brain in one day, according to a back-of-the-envelope calculation that I did, at 100 nanometer voxel size. And a mouse brain would take 7 days, and of course insect brains would be very fast using this technique. And you could potentially even enlarge insect brains more than 4 times, which is sort of the typical expansion microscopy factor. But there's also methods where you can do 10 times or even 20 times larger.
Wow.
And so and of course that increases the imaging time as well because it's a larger volume, but it does make the resolution better if your imaging technique is limited. And so by combining the very rapid scanning, which can be down to 20 seconds even for 1 millimeter cube volume of synchrotron-based X-ray microscopy with the resolution increase of expansion microscopy, you may be able to achieve much more rapid imaging than is possible with electron microscopy, while still getting sufficient resolution to reconstruct the connectome. Now, there's some additional technical hurdles which need to be overcome here, which involve nanotechnology, targeting nanoparticles to synapses, and sort of using both gold and silver nanoparticles, or other. Basically, the important thing there is that you can actually do a dual energy X-ray scan where the X-rays of different energies are absorbed maximally by different elements, and so you can actually get multiple colors by doing X-ray imaging at multiple energies. And the thing is, you have to have 2 different materials which absorb X-rays, and those materials have to have substantially different numbers of electrons. And gold and silver are something, I don't remember the exact number, but it's something like 20 electrons apart or something. And so and there are readily available gold and silver nanoparticles which can be used for these kinds of purposes. And so I guess that sort of addresses your question about nanotechnology, because a lot of these molecular probes, which are able to target specific structures such as synapses and even specific gene expressions, such as I actually did some X-ray microscopy of a rat striatum sample, and that's part of the brain that's involved in addiction and reward. And in this sample, I used gold nanoparticles that were connected to antibodies which target the dopamine 1 receptor, which is a receptor for dopamine, and it is involved in some of these addiction and reward processes. And then I was able to get a 3D reconstruction of the neurons based on where the dopamine 1 receptor was expressed on their membranes. And so this kind of thing, if you're able to do multicolor X-ray imaging, you may also be able to mark where the synapses are, and in that way be able to sort of reconstruct a full connectome using X-ray based technology. And I mean, there's no doubt going to continue to be changes and improvements in X-ray technology. There's actually some even faster synchrotrons which are coming out, 4th generation synchrotrons as they're called, and these may be very helpful in imaging large tissue samples very rapidly. And so I'm pretty excited about that. There's also what's called barcoding, which is actually a sort of an in situ sequencing based method of connectomics. And in barcoding, essentially what happens is you're able to use fluorescently marked nucleotides to take a sequence of images, and each neuron will express a unique, like let's say that. And there's a lot of different variations here, so I'm just going to simplify it a little. Let's say that you have each neuron express a random RNA that has a specific sequence of nucleotides, which are the bases in the genetic code. And this RNA is each neuron expresses a different RNA that's 30 nucleotides long. Now, what you do then is essentially you fix the tissue in a gel, and then you wash it with fluorescently labeled A's, T's, C's, and G's, or U's instead of T's if you're doing RNA, which I guess you'd probably use uracil instead of thymine. But anyway, you'd wash it with the 4 nucleotides, each labeled with a differently colored fluorophore. And what you do is you would, there's some more complicated schemes for how to do this efficiently, but essentially you wash the sample with 1 nucleotide, say adenine, labeled with maybe green, and then you see where in the sample there's a green light that lights up, and then you wash it with say uracil, maybe you've labeled that as red, you see where there's a red light that lights up, and then by taking a sequence of images each time, you're able to essentially generate a barcode, a sequence that is associated with each neuron. And because each neuron expresses a unique RNA, that aids in the segmentation of the neurons, the identification of which neuron is which, and so you can image at a much lower resolution while also having a much higher confidence in the segmentation, since you know that each neuron has this sequence of, say, 18. You would have a unique sequence for every single neuron in the brain. And then in that way, you're able to potentially image an entire. In that way, you're potentially able to image more rapidly as well, because you can use stuff like parallel light sheet microscopes, which are much faster than electron microscopes. Not as fast as synchrotrons, but if you had a lot of light sheet microscopes, which is more feasible than having a lot of synchrotrons, you could probably pull this off. And you could also gain molecular information because you're using fluorophores. You could potentially even simultaneously assay the gene expression of every cell by sequencing other RNAs besides the barcode RNA that you've been using. And so there's sort of this very new sequencing-based connectomics field that's emerging that I see as a very promising emerging technology.
Fantastic. Well, that's really exciting. Ben, did you have any questions or comments on imaging?
Yes. Well, I wanted to take a second and say something in praise of Logan's insect brain emulation proposal, which is, towards the end of the Second World War, there were a group of engineers in Southern California, and in a certain light, those gentlemen were doing high-end work. But they had come from they had built a discipline that had not existed, and in some ways they were a lot of pyromaniacs and model enthusiasts. But they built they'd done the good work of the research, and they were able to pitch to the United States government an framework of an intercontinental ballistic missile system. And out of this small seed grew the Jet Propulsion Laboratory. And I would say that this prospect of insect brain emulation holds similar promise. And it's silly to overlook these ideas. In the pre-interview, we discussed the notion that perhaps synthetic biology and AI research should become disciplines that might merge and might be better used, because the idea of an algorithm being smart, the idea of an algorithm being lifelike, these are good things, but the idea of an algorithm being strong and useful, these perhaps are better things. I'll close by saying that just in recent days, Ben Goertzel gave an interesting speech critical of the state of advanced algorithms generally, called "Are We Building a Psychopathic Idiot Savant Global Brain?" And we need to find ways to make sure that the marketplace and also all of our institutions are able to make a good use and put what is necessary into advancing the aims of folks like Logan. Logan, do you have any? Would you have any suggestions on these related disciplines and how they could advance some AI takeoff effects or more useful products for the marketplace?
Yeah, so I think that the prospects of the prospects here are really about interdisciplinarity and sort of the merging of different fields and the crossover of people with expertise in different fields to gain a much more a much more cross-pollinating kind of environment. And so with insect brain emulation, I think that it's not just about leading towards whole brain emulation of humans and some of the farther off possibilities there, but it's also about sort of this, it's about attracting a community of people together who are from very different fields and who can offer a lot of contributions, and then using that community to sort of spin off a lot of new innovations which could very positively affect our world. So for instance, as you mentioned, synthetic biology, nanotechnology, nanotechnology and synthetic biology could certainly be involved in developing the necessary ways of mapping the insect brain in sufficient detail that you're able to create a detailed biological simulation, and then and the increased flow of resources into these areas may help to further increase their foothold in the marketplace. Furthermore, I think that insect brain emulation itself, as sort of this integrative biological simulation, can serve as a very powerful platform for biotechnological development. And so, if you want to, and it may not just be the insect brain, but it may sort of lead to other types of integrative biological simulations, which are very detailed, and these types of simulations may serve as platforms for the development of a variety of innovations. So, for instance, I can imagine the idea of having a computational simulation of a brain, and then, or a computational simulation of an insect brain, or even of a portion of tissue from mouse cortex or what have you, and then using that to inform our understanding of the neural wiring to the degree that we're able to create 3D bioprinted neural prostheses, which are actually built of real neurons as opposed to electronics. And don't get me wrong, I'm also interested in the idea of electronic implants which emulate areas of neural tissue. But in this particular scenario, I'm sort of talking about how the insect brain emulation may lead to may crossover with tissue engineering, and this sort of crossbreed between computational neuroscience and tissue engineering could lead to both therapeutics and enhancements which could be very beneficial to society. And so, yeah, I mean, I can imagine this world where we're able to map neural tissue, we're able to map how it works, how it functions, and and using that knowledge, we're able to construct very detailed tissue replacements for dealing with traumatic brain injury, for enhancing human intelligence, for fixing parts of the brain that may be damaged in other ways, et cetera, et cetera. And I mean, that's just one example among many possible things.
It's a less invasive Neuralink, basically. Once you it's like reversing Neuralink. You know, I have an interest. You've given me an idea, Logan. I should set up an American ETF on the exchange, just stacked with these companies that are doing this sort of ethical progress. All right, so people can just invest in these companies, and you can get that on an ETF on an exchange, and so you can get the outcomes as they produce products, and also the inputs as they research science. I see this as a huge thing, and if you get the collection of companies that are doing that, I mean that's like, for an entrepreneurial type, that's a self-perpetuating cash cow that could keep these projects engaged forever. But actually, it self-perpetuates into an ethical exchange, because that's what the basis of the ETF would be. Sometimes you're not always going to get your funding from research grants. You have to fight those. You have to rely on politicians and things. But that's super interesting. Or otherwise, they end up in DARPA projects or in deep government projects where they get lost or misused or simply cancelled because of budget cuts or hidden away during in the dust of history. Yeah, really, really cool stuff. Extremely interesting. So, what is the state of research in this field?
Which field? Sorry, are you talking about imaging? Or are you talking about computational neuroscience?
Yeah, neuroscience. Yeah, the state of research, and how would we implement it?
So with regards to sort of these computational neuroscience areas, I think there's several large scale projects, medium to large scale projects that are out there that really exemplify the types of things that we're discussing here. And we've already talked about a couple of them. We've talked about the Human Brain Project and the Openworm Project. The Human Brain Project is a much larger project. Which it actually, I'll give you a little bit more background on it. So it started out with what was called the Blue Brain Project, and they did a simulation of a rat. I believe it was rat, actually, not mouse. But anyway, I think it was rat anyway. Of a cortical column. From a rat, which is essentially a part of the cortex, which is in some way structurally self-contained. Obviously, it has a huge amount of communication with the rest of the brain, but it sort of operates as a repeating unit, which is seen across the cortex. And there's some level of self-similarity between these repeating units, though obviously there is a great deal of diversity between them as well. And so this cortical column simulation was essentially a first step that the Blue Brain Project took, and the thing was, though, that it was informed more by these approximate wiring rules, which I think was what you were asking about earlier. Can we algorithmically generate a connectome? And I think we actually forgot to go over that part of your question, so I'll address it here. We can, to some degree, algorithmically generate neuronal connectivity and morphology. Well, actually, we usually image the morphology, and then what's done is we use other data about connectivity patterns to sort of insert those morphological reconstructions into 3D space and then wire them together in a way that approximates what might be found in a brain. And that was what was done in the Blue Brain Project's cortical column simulation. And this was a good step, but it ended up being that the larger neuroscience community was a little concerned that because it was so approximated, it ended up being some concern as to the usability of this simulation, as well as because it was a fairly small part of a larger system. And so there ended up being some shifts in the organization of the Blue Brain Project, which was later called the Human Brain Project, which led to more of less of a computational neuroscience as its sole focus, and more of a hybrid of experimental neuroscience, computational neuroscience, and working towards applications. And so in the realm of, and I think that that's good, because I mean, they were, as I mentioned, Henry Markram, who's the founder of the project, did sort of propose human brain emulation by 2020, which was quite premature. And so I think it is good that the project has sort of shifted to some degree. However, the Blue Brain Project does still exist. It's sort of a subcomponent of the Human Brain Project, and the Blue Brain Project does still aim towards these more computational neuroscience integrative simulation types of goals, though they aren't, I don't believe that they're aiming to do whole brain simulations of humans in the near future. I do see it as sort of a sort of a foundational effort in that eventual goal. It is one step along the way. There's also the Openworm project, in which the 302 neurons of the C. elegans worm, which is a type of worm, it's not like an earthworm. It's nearly microscopic. It's very small. It has 302 neurons. I think something like 7,000 synapses. can it be 7,000? I don't know. Anyway, it's a very simple organism, but the Openworm project is sort of an open source project where a lot of people have volunteered to use what has been gathered about the C. elegans connectome, which the C. elegans connectome is known at this point, at least for two worms. There was a recent study where they got both a male and a female. Originally, there was actually a connectome available as early as the 1980s of the C. elegans worm, but it was only for a male, I believe. And so, although actually I don't remember which one it was, but it was only for one of the genders. I'm sorry. It was only for I believe now it's a male and a hermaphrodite. Anyway, sorry, I'm a little rusty on that, but the details aren't really all that important. It's just multiple genders of the worm. There was a recent study where they got connectomes for both, and so they've been using these data as well as a lot of other data from like gene expression atlases and so on and so forth from C. elegans to reconstruct a virtual C. elegans, and they actually have sort of the nervous system of the C. elegans all put together, but they don't have the muscular system fully linked into that nervous system, and so they're currently working on that. They're also working on a fluid virtual environment where they use fluid dynamic simulations to create a virtual environment for this worm. And then they're going to have sort of sensory feedback as well, where the virtual environment will give the worm sensory feedback. So it's a bidirectional loop between the virtual worm and its virtual environment, and we can learn from that. And so that's currently what's being worked on with that. And with regards to other projects, there's a lot of, as I mentioned earlier, there's a lot of electron microscopy work being done at Janelia. There's a lot of electron microscopy work being done at the Allen Institute, as well as at Princeton University and Harvard University. And then there's some more emerging technologies coming out of places like Cold Spring Harbor, where the sequencing barcoding stuff was born, and also there's a fellow named Bobby Kasturi who I've communicated with, who is doing some synchrotron-based work for X-ray imaging of brain tissue, and then there's me. And there's a few others as well across the world who are doing this sort of the emerging technologies for connectomics kind of research. And so yeah, that's sort of an overview of what I see as some of the more important research going on that's relevant to these kinds of computational neuroscience goals. One thing I should mention is that the image processing component is actually a fairly major hurdle to get through. And so there are people like Sebastian Sung, who is at Princeton University, as well as I believe his name's Moritz Helmstadter, who's somewhere in Europe, and there are a variety of other groups who are, there's a couple of groups at Harvard that do computational processing of connectomics data, and so trying to take these very complicated images that have very dense neural tissue, and to distinguish which neuron is which is a fairly major computational hurdle, although there has been very major progress that has been made in over the past several years even, and that progress seems to be accelerating. And actually, when I was speaking with Sebastian, who's at Princeton, he seems to think that based on his current research, that a lot of the challenges that have been fairly prohibitive at this point are going to be overcome in the coming few years. And so, and there have been some other opinions where I believe Helmstadter had a paper out where he was saying, oh, well, it would take a lot to reach that point. But I have heard from some that Sebastian's group is likely fairly far ahead of Helmstadter's group, but I can't really speak to the details of that because I haven't been involved in either of those groups extensively. I've spoken with Sebastian and I've gone to some of his lab meetings, but I haven't been in the groups for. I can't really compare for sure, but that's just what I've heard. So the computational segmentation people are also a important component of the current state of research. So hopefully that sort of answers your question about what the important technologies and approaches that are out there currently are with relevance to this area.
Ben Zion, you were going to say something about the worm?
Well, I actually wanted to ask Logan a question about tech in China. It's probably accurate. It's not wholly inaccurate to say that the computing power required to do an insect brain emulation, the access to that could derive from the kind of techno-nationalist institutions, a kind of state-run tech in China. And also, I think it relates to other projects that we've talked about here. There is a China brain project, if I'm not mistaken. Would you want to go and work on that? What's the state of that? It's a little opaque. Could you tell us a little bit of that and criticisms of this relative to the well-known criticisms that others have made of the human brain project? And China. Go.
Yeah. So in China, you're right. There is a China brain project. There's actually also a Japanese brain project, and I think there might even be one in Australia. Not super familiar with that one, but so there's the China brain project. There's Japan's brain/minds project. And so there's a lot of these more Eastern approaches or like these Eastern run initiatives towards understanding brains that are sort of similarly large, maybe not quite as large as the European Human Brain Project, but or the U.S. Brain Initiative, but there are these Asian initiatives where they are working on some fairly large-scale neuroscience. And what it seems to me is that a lot of the stuff going on in those places is more oriented towards clinical neuroscience. And I mean, it's not all, but there does seem to be less of a focus on the computational principles of how brains work and more of a focus on just sort of like what can we use in the near term. So for instance, with Japan's Brain Minds project, they're using marmosets and they're building a mesoscale connectivity atlas of the marmoset brain, which I think is very valuable for these more clinical near-term applications, but maybe even is valuable for weak artificial intelligence, but may not be as useful for really understanding the computational principles of the brain to the degree that we could use it to develop strong artificial intelligence or even maybe inform robotics. And so I think that those projects are definitely very valuable, but I'm not sure if they're as relevant to what's going on with whole brain emulation. Nonetheless, I don't know as much about those projects, so it is definitely possible that I'm missing something here. I do seem to recall that there was, when I was reading a paper on the China Brain Project, there was mention of sort of an arm of the project that was focused on artificial intelligence, and so that's something worth investigating more deeply. It's interesting. I haven't heard as much about
largely public health focused.
Yeah.
So we all made a reference to the sort of the Ben Goertzels and Ray Kurzweil's working on advanced algorithms, and the Elon Musk's working on brain-computer interfaces. In your view, which of these two in this arms race is more likely to sooner produce a functional superintelligence? And would you consider these kind of brain-computer interfaces if they were somewhat invasive but provided those benefits?
So I don't think those kinds of brain-computer interfaces are necessarily going to produce a superintelligence in the near term. With regards to who might win the superintelligence arms race, I actually think that by the time we get to the point where superintelligence becomes more of a real possibility, I think that the world's landscape may look different than it does currently. And sort of in a best case scenario, I can imagine that there would be a greater degree of communication between nations, a greater degree of international collaboration, where we would all sort of together win the arms race instead of sort of competing separately. With regards to like if you just strictly look at right now, I actually think more the European, United States types of crowds are seem like they're more moving in the direction of artificial superintelligence. However, there is some interest in that in the more eastern areas. Masayoshi Son is a businessman who is in charge of the company SoftBank, and he has actually created something called the SoftBank Vision Fund, which is aiming towards eventually helping facilitate the development of artificial superintelligence. But again, that the Vision Fund is collecting investments from a large number of companies, not only SoftBank itself, but also I think the second largest investment, or possibly even the largest investment, was from a Saudi Arabian company, or maybe from a Saudi Arabian prince of some kind. And then there's been investments from a variety of other groups sort of across the world. And so I do kind of think that it's, although the Western areas seem to be more involved in thinking about artificial and superintelligence, there's certainly some thought in some of the Eastern areas towards that idea. And I think that, but I do think that it's likely that in order to construct something, to undertake a project of that magnitude, I think that there will need to be international collaboration between these different parts of the world.
Yes. I pardon me.
Are you dying?
Yeah, we were talking in the pre-interview about my death. We were talking in the pre-interview a little bit about that sort of, in a word, the Chinese techno-nationalism, this cultural interest in when DeepMind, when this AI beat the Go champion, many tens of millions of Chinese and East Asian folks began enrolling. There was a sharp uptick in enrollment in computer science courses, and this does seem to be something that's rather lacking in the United States. And it's interesting, I think, that we could see so many new cultural properties and institutions besides directly deriving from advanced algorithms that change the face of the earth. This is something that.
So Logan, you're talking about this horizontal integration, through something like a mega bank like SoftBank. Do you see legislative issues that on the horizon, things like the United States Transhumanist-Party, ZION2020, we are Zoltan Itzvan, for example. Do you see issues that need to be addressed, that need to be talked about in the public space, to do with the research, to do with the imaging technology, to do with implementation and the ethical and societal sides? The debates that are being left out in just omitted from public discussion, and if you do see them, what should we be talking about? How should we be talking about it? And especially on this horizontal integration and collaboration. But anything you like, what would you like to see more of in the mainstream media, in political discussions, etc., talks between friends, etc.?
Sure, yeah. Before I answer that, I'd just like to go back to what Ben was saying, and I do think Ben certainly seems to have more of a focus on international politics than myself, and so I definitely do see that there might be some emerging Eastern interest in this area, and that could lead to sort of an explosive growth in the amount of innovation coming out of those areas, and partially perhaps the reason that I haven't heard as much about transhumanist or whole brain emulation types of thought coming out of Asian areas might just be because I live in the United States, and so I hear more about this type of thing. So I definitely am less on the policy end of things myself, but I do like to think about these topics, and I feel that that it is a very interesting area that I hope to continue to learn about. Going back to your question, there's what were you asking again? You were asking about sort of the
what's the emission?
what would I
What's
like to see in
emitted
mainstream
from
media?
what's emitted from societal discussions that needs to be talked about now by politicians and by people in the community and business leaders, etc. To do, especially to do with collaboration, horizontal integration, things like the Human Brain Project, because these are these innovations. They're super big. They're super quick and they're changing society, but we don't hear this sort of discussion anywhere.
Yeah, so I think that I do think that there needs to be sort of more of a public awareness of where the future is going. And I mean, there is some of that at this point, but I do think that it's a very superficial coverage. So you'll hear headlines about artificial intelligence and synthetic biology, but the media seems to oftentimes take very oversimplified and oftentimes sensationalist, usually pessimistic sensationalist kinds of approaches to these to these topics, and so the public just ends up getting these sound bites, which are usually playing on their fears, and you end up hearing and then there ends up being sort of a negative public opinion, which can stymie the progress of large-scale projects, which do need to have some involvement of governmental entities. I mean, there definitely is the possibility of commercial projects as well, but because some of the outcomes here are not direct, it oftentimes ends up being that there needs to be sort of both governmental and commercial entities involved in these projects. And so when you have this negative public opinion of science and technology, there ends up being sort of the danger of that leading to people being too alarmist and trying to stifle and ending up stifling some of the integration of these fields. And I mean, that's not to say that I don't think that there should be any caution, because I mean, I absolutely agree that there are risks to technologies, and there are things that we need to think about ethically. But I think that oftentimes what happens is that people end up getting sort of caught up in alarmist thought, which is not necessarily addressing the real ethical issues of a technology, and more addressing sort of the disgust reaction to something new. So, for instance, with things like with things like whole brain emulation, I mean, people might be afraid of it because because they're worried about sort of like this Terminator scenario or something, whereas really there's much less risk of that than there is of creating a entity that could be conscious and could be subjected to suffering in experiments. And so I think that we sort of need to shift our ethical focus from the really grabby, alarmist kind of stuff to what could really cause harm and what could cause benefit. Another area that I think doesn't receive enough attention is animal rights. And so, I mean, obviously animals do need to be used in research, but things like the rights of animals, things like the rights of virtual entities, things like the rights of cerebral organoids. I mean, I think we do need to have an ethical discussion around what could be causing harm and how to best mitigate this harm while still allowing research to progress. And so, yeah, I guess that's another thing. Just the idea of suffering as sort of something that we should pay more attention to in ethics, I think is especially important, because as we're talking about engineering intelligent entities, I mean, we're going to have to worry eventually about this idea of like, what if they do suffer? What if we're creating something and then we're using, like, what if we create a human brain emulation and then we use it to study mental illness and we put it through a whole bunch of mental illnesses? That would be bad. And so but that's a little bit less grabby than like the Terminator scenario or what have you. So it's I think that we sort of need to understand that some of the ethical issues can be a little bit more subtle, and we need to sort of shift the direction of the conversation from our sort of instinctive reactions to change and more to directly addressing what causes suffering and what causes a reduction in suffering.
Ben, did you want to comment on that?
Well, no, that I agree. I agree with all of that. I was going to say a little something else about the transhumanists that we know, in contrast with emerging culture and very real institutions in East Asia. Japan doesn't have a transhumanist party, and maybe in terms of life extension, they might be in some ways better for that. The transhumanist cause, it is very interdisciplinary, and there's a kind of techno-fatalism in that. These things will come, and that's something the Japanese have a culture of interest in longer lifespans. They have the investment in that. The Chinese have and as well this kind of interest, but this parallel interest in advanced algorithms and this work toward that. The most important thing is really the funding of these things, the creation of institutions and cultural properties, and it is concerning, for example, that we might in attempting to create we might create something that we are subjecting to untold horrors in attempting to create in silico life, but in almost all of these scenarios, I think it's really best to focus on the raw tonnage of computing power that derives from from trying to find the best ways toward innovation in these areas.
Yeah, there was a question about quantum computing, and do you think quantum computing will accelerate any of the advances, whether that's imaging or the algorithm idea of skipping the research, or data mining, or anything really to do with the actual research side? Will quantum computing have an impact, and if so, how substantially?
I think that, quantum computing will probably have an impact on computational neuroscience. Quantum computing is actually an area I don't really know a huge amount about. However, I do know that one of the applications that quantum computing is very good at is computational chemistry. And so I think that in particular, if we do need to incorporate more detail into our computational models, and include a subset of some of the chemical processes which are occurring in neurons, we may be able to benefit from from quantum computing there in particular. It could also greatly aid in our understanding of the computational chemistry aspect of quantum computing could also greatly aid in our development of nanotechnologies, which could support better imaging processes, better molecular probes, better ways of understanding the nervous system in general. And so I think that quantum computing will, when it comes to understanding chemistry, be of particular impact. I can't necessarily speak as much to how it will impact the simulations themselves, Although from what I've heard, I do suspect that there may be ways of applying quantum computing such that it would be able to accelerate biological simulations. And so, yeah, I think that, I mean, like neuromorphic computing, quantum computing is sort of a specialized architecture which is very good at certain tasks. And so I think that if we're able to sort of craft our computational neuroscience such that we're able to take advantage of the tasks that quantum computing is very good at, we'll be able to make some great strides using that. But again, I don't necessarily know all the details of that, of how quantum computing may contribute, but it's definitely a very interesting area, and I think that there's a huge amount of potential.
Wow, cool. So what does all this mean for society as a general broad question? What does all this mean for society, Logan?
So I think that there's sort of two different segments to this when it comes to society. There's the short term, which is, I think what it means essentially is better biomedical technologies, better understanding of biology as a system, better synthetic biology, better nanotechnology, ways of interfacing with biology to sort of bring us closer to our technology, and in that way bring us closer to nature, in the sense that we're able to more seamlessly and smoothly interact with the world around us. And so I think that there's going to be sort of this influx of nearer term changes, which are just essentially making society better, making making us healthier, making it so that we can live longer, making it so that we are able to overcome more maladies. And as well as helping us in our general design of weak artificial intelligence, and our general design of algorithms in computer science in general. I said general too many times. In electrical engineering, and actually that's one thing that I think might be of interest, is that neuromorphic architectures tend to be very low power, and so if we're able to gain more of an understanding of neuroscience, we may be able to further take advantage of some of the low power properties of neurobiological tissue function and implement that in our electronics, thus leading to more energy efficient electronics and less impact on the environment. And so I think that there's going to be sort of this nearer term stuff where we see a lot of really positive effects. And then in the longer term, I think that there's going to be more there could be some very, very good things that could happen, or some very, very bad things that could happen. And I think that the good things are good enough that it's worth striving towards and taking the risk, and trying to approach it in a way that we do reach the good stuff and we minimize the possibility of the bad stuff. And basically by that, I am talking about things like artificial superintelligence, things like copies of humans. I'm expecting it to be. I think it's kind of unlikely that we could, in the next 100 years or so, reconstruct the connectome of a living person, but I think that a person who has already died, we could potentially within 100 years, maybe 150, we could essentially create a copy of that person in silico. And I sort of envision this world where consciousness and memories are sort of almost like files on a computer, and we're able to move them around and duplicate them and so on and so forth. And I think that has huge potential for a lot of beneficial things, but also huge potential for some negative things, like when it comes to suffering. So I think that there will need to be an increase in cybersecurity before that kind of thing would become widespread. And with regards to the positives of that, I think that I sort of envision, I don't know if you guys have heard of von Neumann machines, but sort of this idea of self-replicating spacecraft that may carry a virtual consciousness and can go out into the universe and seed the universe with life. I see I sort of see the idea of creating copies of people and instantiating them in silico as a way of perpetuating the human legacy, of making it so that we don't die out in the next 10,000 years or whatever, or in the next 100,000 years even, but rather we're able to sort of transition our culture, our emotions, our souls in a way into a computational substrate, which is much more able to spread across the universe, is more suited to surviving in outer space, and can directly interface with technology in such a way that it could manipulate matter to sort of self-replicate on other worlds, go out there and allow us to keep our humanity going, as well as to develop further into post-human life forms. But at the same time, sort of maintaining some of the good essential components of what we consider to be our human legacy. And so in the longer term, I see that as the sort of grandiose vision of what could potentially happen if we play our cards right. And I see that as very worth striving for. And I mean, from there, you can imagine all kinds of like cosmic civilization stuff happening. And there could be some very interesting things that would come out of that. But I do think that we need to base our what we design as essentially as our virtual offspring in a way. I think we need to base that in biology, because if we try to develop artificial intelligence without it being based in biology, it may have very alien motivations, very alien ways of working with the world that we don't necessarily want to be spreading across the universe. We want certain human values to be maintained. And I think that although there are many flaws with current human society, there are also a lot of things that would be worth carrying on into the next stage of our evolution. And so I think that it is important to use bio-inspired technology rather than trying to de novo invent artificial intelligence, because if we use technology that is based in connectomics, we can actually maintain the sort of core human values, the core things that we feel are important to carry on into the future.
You know, those two models bring to mind a serious problem with this as a life extension strategy. You know, there's relatively few technologies that human beings don't try to develop, and a lot of those concerns about those technologies they derive from security or privacy concerns. I won't go off on this, but you could watch the show Steele did last night with Kristen Conklin that describes some of those kind of prospects. But what I'm getting at here is just the cost of life extension. So a brain transplant, cloning myself and putting that if we're talking about a commensurate AI development that allows for life extension, we're talking about $1 trillion+ of R&D. If we're talking about me having a physical immortality through even something as outlandish as that, which is maybe not the lowest hanging fruit for longer lifespans, but even that, if somebody said, here's $1 billion, Mr. Caballero, or the Chinese scientist who's better regarded than that guy, you would be able to, and it wasn't made completely illegal, you would be able to, I believe, develop a protocol for brain transplantation in a period of years if the money was there and people were not completely losing their minds about the idea. But the ideas of doing this, it is indeed a longer term consideration, as you said, because we don't know where or when we'll see these takeoff effects, but it does seem that absent a dramatic change in the way the economy is structured and a sort of economic singularity, that that my prospects for life extension deriving from AI would not be enhanced without somebody spending more money than anybody can really marshal at this time.
So yeah, I actually I think that the prospects for gradual replacement mind uploading within our lifetimes is actually rather poor. So I actually don't think that, for me, individual life extension isn't necessarily the top priority. I think that it would be great to live forever, and I'd really love to see the future and see what happens. But I sort of see the preservation of the human species as sort of a something that would come first. And so I'm personally okay with the idea of potentially living for like a few hundred years or something like that, with the help of molecular gerontology, and then having something, and then having some sort of mind clone live longer. And so I don't necessarily see the idea that the sort of scan and copy mind uploading idea is necessarily, the fact that it would be just a copy isn't necessarily bad. I mean, it's not as good as it would be if it was me, but I sort of, I mean, it's like, in the grand scheme of things, okay, so there's a copy of me that lives forever and I don't live forever. I mean, the copy gets to live forever, and then it goes on and continues to do stuff. And I mean, there's other people who may be copies and will go on and live forever. And from there's going to be a much easier transition into further developments. And so I don't necessarily see that as a huge negative. I mean, like I said, I'd love to live much longer than that, but I sort of put the priority of keeping the human race going before the individual life extension stuff. I mean, I think that it's definitely possible that there could eventually be sort of this more dramatic kind of life extension from mind uploading, and I'm definitely not ruling that out. But I think that in the next couple hundred years, it's kind of unlikely that would happen. But there could definitely be that possibility farther out. There might be, and I mean, the future could be very strange as well. I mean, we may not be able to predict all the things that will be able to happen. I mean, of course, we wouldn't be able to predict everything. So, I mean, there are some possibilities out there, but that's just sort of my take on it.
You know, there's just,
Ben,
you probably got a better question. You keep talking.
Oh yeah. Well, what you said about the before about the mouse brain emulation, I think it promises to answer some interesting questions here of what we've just described. You know, I'm not inclined to think that emulations in any near-term context are going to have the experience of emotional or physical pain, or really experience human motivations or experiences to any high degree. But that mouse brain emulation, when it does have the R&D behind it, could maybe start to answer some questions of this kind. I guess that question does lead back to a concern that you described. It's true. I want life extension for myself and for other human beings, and there's better ways than wild psychosurgery to do that by the present estimation. But it does seem possible that we might not quite be able to preserve humans in the foreseeable future. I hate that term. In emulations, because we don't really know what we don't really. There's an awful lot of parts of the equation that we might not even be looking at there for the creation of experience. not embodied experience.
Right. So yeah, I agree with that, because I mean, I think that's definitely a challenge that we would have to overcome. I don't think it's an insurmountable challenge. Like you said, the something like a mouse brain emulation might help us start to answer some of those questions. I think another thing that's important to consider is neural prostheses. Because if we have, like, let's say that there's somebody who has a big chunk of their brain taken out in a motorcycle accident or something, and then you give that person a neural prosthesis which very closely emulates the part of the brain that he's missing. Does he or she still have this? Does this person still have a conscious experience through this neural prosthesis? And I mean, and I think it'll be sort of a process of trial and error to figure out what types of architectures may best give rise to something that would be like an embodied experience. But I think that analyzing it through sort of human-machine hybrids, which, of course, as we mentioned earlier, is sort of being worked on at Neuralink to some degree. There's also Theodore Berger's hippocampal prosthesis, which is sort of an early version of kind of what I was talking about there. And so I think that to answer these questions of whether a emulation will be conscious, I think there's going to be some of these biomedical implants will definitely be very helpful in the philosophical analysis of that, and sort of help us to better ground the philosophy in data, and move between sort of the fields of understanding consciousness at a very abstract level and understanding consciousness in a more data-driven, concrete level. And so I do think that it's something that it's possible that we can figure out how to engineer physical matter in such a way that real conscious experience does emerge, and that we can verify that. But I think that it's definitely going to be another engineering challenge along the way.
Mm-hmm. And I'll say of this exobrain notion, you and I got a chance to speak for a few moments to the author of Artificial You, AI and the Future of Your Mind, Susan Schneider, and this is a really good book, and folks should read it. But it does do a certain kind of a concern trolling about this broader exobrain phenomenon. And I would like to again encourage people not to be concern trolls, but to embrace the use of these technologies. They do potentially have personal and national security implications, and those are well articulated in this book. But really, the bigger issue, in my view, is that these things could dramatically improve our quality of life as a species.
Absolutely.
Sorry to steal it.
No, that's good. That's really comprehensive questions and answers, and I'm really enjoying this line of thought. I was just going to say before, there's this dystopian idea of this episode. I haven't actually seen it, but I've read a synopsis of it. Monkey Needs a Hug from Black Mirror. Have you seen that, Logan?
I have not seen that one. I've only seen a couple of Black Mirror episodes. San Junipero and the Entire History of You.
Okay, cool. Ben, you're laughing. You know the episode I'm talking to about?
Yeah, I think so.
So basically, the synopsis here
I've seen them all. I'm trying to recall.
The synopsis is here. Let me just read this so I don't get it too, because I haven't actually seen the episode. So this is from theringer.com/TV. The episode is called Monkey Needs a Hug. The rundown: new technology makes it possible to transfer one's consciousness from entity to entity. In Black Museum, a comatose woman's consciousness is first transferred to her husband. However, when he gets tired of hearing her in his head and wants to move on with another woman, he transfers her consciousness to a toy monkey. A presentation: the monkey can only say two things: monkey loves you and monkey needs a hug. Deadliness. They are not lethal. There are no lethal consequences here, only existential. The monkey can't even move. Not very deadly. It is, however, very sad because the monkey is tossed aside and eventually thrown into a high-tech crime museum with the woman's
That was an
consciousness
anthology episode.
still
yeah.
inside of it. Here it says, "Would this ruin society?" But we don't need to read that. But what do you say of this idea? You know, you've got a mother. She's in comatose. She wants to see her son grow up, and be with her family. Her consciousness is now uploaded to her husband's head or some object like a monkey, for example. And now she's left in a crime museum. Is this a worry? Is this sort of stuff a worry? Again, you don't think this is on the horizon for the next 100 years anyway, right? But we've got Mr. Mike DeVerde, superfan Mike DeVerde in the comments. He said something very interesting here. Let me just pull that up. We love superfan Mike DeVerde.
Let me just say
He said linear progress might indicate hundreds of years for WBE, but exponential progress may lead to WBE by 2050. Logan, what do you say? Do you see 2050? And also, what about this monkey needs a hug idea of existential dread?
Yeah, so I think that there is exponential progress, and that's sort of what I've been talking about with this idea of different fields converging and enhancing each other. So fields like nanotechnology enhancing neuroimaging, and then that enhancing computational neuroscience, and then computational neuroscience feeding back into electrical engineering and artificial intelligence. And so I think that there does end up being an exponential growth in technology. However, I think that personally, from what I know about the brain and about how the brain interacts with the body as well, which is a very important component. I think that I mean, even with an exponential growth, it still would take quite a while. And so I think that it's sort of like you have a very fast growing function that's trying to reach a very, very high number. And so the fast growing function will reach the high number eventually, but even with that fast growth, it may take some substantial amount of time before it hits that point, just because of the sheer complexity of the problem. And so I think that there is definitely an exponential growth, and I think without the exponential growth, it would take perhaps tens of thousands of years to reach this kind of goal, whereas I'm estimating closer to a few hundred years. But with the exponential growth, I think that the goal actually does become tractable.
Cool. And the monkey needs a hug.
I'd also like to interject that by the sort of we spoke of Susan Schneider's work and this exobrain. Even by the prototypes of today, I think there are senses in which I am now a monkey who needs a hug relative to my exobrain improved self. And by the prototypes of 20 years from now, I'm certain of it. It's fine to concern yourself with exotic thought experiments about worst case scenarios, and that's a sort of a wonderful thing in fiction, and it's right, but it's a little bit. It's also I think yeah, I like I do like that exobrain technology. I want some Neuralace right now.
Yeah. So yeah, to answer your question about the monkey needs a hug, I think that the idea that a artificial consciousness would be vulnerable to being trapped somewhere is definitely a concern. And for that reason, I think that it would be, my hope is that there will sort of be a parallel development in technologies which allow us to sort of almost automatically correct for and have fail-safes for that kind of scenario, preferably many layered fail-safes, so that there's some degree of redundancy in case some of the fail-safes themselves fail. And so I do think that sort of the idea of a virtual mind being trapped in a black room or in a toy monkey or something like that is something that we will have to be cautious of, but I don't think that it's necessarily impossible to avoid that from happening by simply implementing the proper safety measures. Just
So
like,
it's just
I
an
mean,
advance in cybersecurity measures. The other issue I wanted to ask you, you touched on this very interesting point before about the fact that biologically we should be integrating with the computer algorithm in order to keep some humanity or what the values that make us human intertwined with the silicon the new race, essentially the new advanced the species. I was wondering on your analysis or what you think of as actually being human, as like what do you actually see in humanity, and what do you think? Do we need to do more research on actually what makes us human? Are we, we're getting there with the body. We're starting to understand the different components, the biological components that make it us. Consciousness is, and the substrate of consciousness is still something that's tricking us up. What do you think personally, or what do you think? What institutional framework do we need to build up to actually really understand who we are before we decide to make this transition? And do you think that this transition is even possible until we understand exactly definitively who we are, or is that a leap of faith? Is that a leap of faith if we don't really understand it and we do the conversion?
well, I personally think that sort of the humanities is a very rich resource for this kind of thing, because I mean that's essentially what the humanities have been studying for thousands of years. Well, maybe 1,000 years. However long the humanities have been around, I mean, these institutions which study human emotion, human culture, the dynamics of human interactions, and so on and so forth. There's a huge amount of existing research into who we are and what makes us human in that sphere. And I think the only missing component there is bridging the gap between the sciences and the humanities, in that there's currently sort of this cultural divide between the two, which leads to a lot of division and a lot of suspicion, particularly when it comes to very sort of science-focused initiatives like transhumanist goals, like the transhumanist goals we've been discussing. There's a lot of potential for mistrust from the people who are in the humanities, and I've seen this in a lot of the literature that I've read from such folks. However, I think that if we are able to have better communication between these two segments of academia, we'll be able to take advantage of the vast and diverse literature of ideas around what really makes us human and what's valuable about being human that is present in the humanities. And I think this is also partly where science fiction comes in, because science fiction really allows us to explore both the sort of our visions and our hopes and dreams for the future, and it allows us to explore the sort of the closer zoomed in human element, which is very important. And so I mean, when I've actually been writing some science fiction lately, and I've been thinking about what about this possibility of, if there's copies of individuals, how will the copies feel knowing that they aren't the original? How will they structure their identities? What will their lives be like? And so I've been sort of exploring that through science fiction, and I think that sort of bringing forward the humanities into the realm of science through writing and through cultural analysis and through digital humanities and through sort of this integration that makes our fields converge more, not just within the sciences, but between the sciences and humanities. I think that we really will be able to access the immense resources that are available there, and bring that forward to allow ourselves to really make informed decisions as to what we want to look like in our future going forward.
Would you think, hypothetically, if this was a simulation that we were living in now and we were simulated, either on our own, like we are our own copy of something. I mean, wouldn't that just be the answer if this was hypothetically that answer? Like if you were a copy and you didn't know, wouldn't you just live the life you're living now, which means that in the 21st century, at this point in time, you are doing that? Isn't that the answer if hypothetically this reality was a simulation? Because you wouldn't be aware. Now, if you were aware, that dynamic would change, right? But hypothetically, isn't that the answer?
Sort of the answer to the ethical problem.
Well, like if this reality is a simulation and you are a copy of yourself, but you didn't know, then therefore that's the answer of how you would live your life, right?
Sure. I mean, you could say that, but I mean, I think in the practical context, there would be a little bit more trouble with. I mean, how would you necessarily, like, if you made you'd have to make like an entire simulated world essentially for the copies, and then sort of put them into that world and have them exclusively live in that world. And so I kind of am thinking more that it would be sort of a more beneficial project to have the copies know who they are, but have volunteered to be to be created in such a way that they are a copy of people who are existing, and then to use those, and then to have those volunteers sort of go out into the universe and seed the universe with life. And in that way, I mean, it wouldn't just be sort of a simulation within our existing world that wouldn't really be doing a whole lot. It would be them sort of actively piloting these von Neumann machines and going out and helping to preserve the legacy of the human species. I also am not entirely sure if I'm comfortable with the idea of creating copies of people and then not telling them that they're copies. Just to be fair, I'm not sure if I'm entirely decided on that question either, but I think it definitely could be interesting to analyze that from sort of a philosophical ethics perspective as to whether it's better to give sort of this informed consent to such entities, or whether it would be better to just allow them to live their lives as if they were the originals. But I mean, again, that's I personally think that it would be sort of practically more important to have them know that they're copies, and then have them just be volunteers in that space.
It's interesting you mention this because, you know, we kind of live in a world which is in the intermediate in that sense. We're in the middle of that range because, you know, if you're a religious person, you're on a transitory journey from one dimension to the other dimension, and you kind of know that you have a copy when you turn into an angel after you die, or whatever your religious belief is, but you also have this fragment of information from the religious texts or from prophets or deities that have come down, such as Jesus Christ or something, who have led these fragments of information that there is another realm, another dimensional spiritual space. So we live in this world where that law is almost smack bang in the middle, and you can really make your own choice on that. And I think that, you know, and that's reflected in a lot of religious beliefs going back to Adam and Eve. And, you know, if you think about the first story, what is it about? It's about eating of the fruit of knowledge, right? And they have a choice whether to be ignorant or whether to to ingest the fruit of knowledge and, you know, go down the path of making decisions. And, you know, and one of the punishments, essentially, if you play that out, could be this sort of quandaries that, okay, now you've played out technology, now you get to be God and have the same problems as the creator or the entities in the prior, you know, fragmented existence in that spiritual realm. So essentially that sort of quandary is kind of a punishment.
There are very small sample size experiments effectively that identical twins separated at birth, unaware of having a twin, and their response to this is not that exciting. that doesn't rock their world. Every one of us is sort of has in the form of our parents, folks from whom we derive, and we can choose to attach a lot of significance to that or not. I don't see this idea of we're kind of getting into some idealistic discussions here. I don't see this a problem of the simulation theory or simulation theory related ideas in advanced algorithms as being very pressing concerns. I mean,
yeah, I mean, I kind of agree with Ben here. I mean, I think that when it comes to this idea of the ethics of having duplicates of individuals, and sort of paralleling that with the idea of simulation theory types of ideas, as well as these religious parables that you've described, I think that I mean, I do think that it is a concern to think about, but I don't think it's necessarily the biggest concern. I mean, I think that some of the other stuff we were talking about might be more pressing matters.
Yeah.
But I mean, like I said, if the people who are duplicates volunteer, I think there might be some amount of internal seeking that must be done by the virtual copies of the people in order to sort of reestablish themselves and their identities. But I don't think that that's necessarily a huge challenge, at least relative to the level of the other challenges. So I kind of agree with Ben here. By the way, I just want to let you know I should probably start wrapping up pretty soon. So if you want to cover anything more in particular, we should probably get to that.
I'll quickly just ask you, what directions can this lead to, like bioengineering, implants, synthetic biology? We'll just quickly go over that.
So I sort of see these integrative simulations as leading to a much better picture of biology as a system, because at this point in time, and especially up until recently, biologists have primarily studied biological systems in a very a very tiny piece of the puzzle kind of context. They've interrogated some protein or some pathway and looked at what it does, and it really intensively analyzed that one thing. And there have been sort of this emergence of more omics types of initiatives, like the Human Genome Project, and like some of these neuroscience initiatives that have come out, where we're trying to analyze biology more as a system. And I think that sort of the convergence of these systems database type generating approaches, where we are gathering these massive amounts of data, is sort of leading us towards this a world where we're able to make much better predictions about biology by integrating these data into simulations. And so I think that with regards to biomedical advancements, there's going to be when it comes to biomedical advancements, I think there's going to be a huge amount of benefit from having simulated organisms, and even just more complicated and more biologically accurate simulations of parts of organisms. I think that there's going to be a much better ability to predict how biological systems will behave, at least to a reasonable degree. And using that predictive ability from these computational models, we're going to be able to do much more elaborate types of biomedical engineering and create much better therapies and even enhancements than we have been able to up until this point, simply because we have a way of very rapidly profiling our predictions in silico and guiding our hypotheses using a systems-wide understanding of how biological organisms work.
All right, that's fascinating, fantastic. Well, if you haven't got anything else to say, Ben, I'll wrap it up. Have you got anything else to say?
No, only that I share this excitement for this emerging discipline as being one that could have tremendous impact on the marketplace and other disciplines. And I would encourage people to continue to support and follow Logan's work and support projects of this kind more broadly. This has been a fascinating conversation. Thank you both for having me. I'm looking at the timer down in the corner, Steele. That's coming very soon, but I think that puts us about 24 hours shy.
Yeah. I don't know the exact time of the Chicago debate, so I guess it's close.
It's close.
It's relatively close.
Yeah, so I want to remind folks, please, if your friends or family are in the Chicago area, come on March 4th at 2 p.m. To the Chicago Hilton, and it's going to be an exciting event, open presidential primary debate, the first of its kind. A lot of press interest going on there. And the Free and Equal Election Foundation has done, again, something unprecedented. You can watch that also at freeandequal.org. And you can check out ZION2020 and learn more about the Futurist New Deal for America.
Yeah, and we're really looking forward to that. It'll be a historic debate, lots and lots of parties there, and US Transhumanist-Party is going to roll them, so that's going to be really fun. Well, hey, look, Logan, thank you so much. You're incredible, great mind. Your work and research in the description. We'll link that in the description so people can have a further look at that. All the best with Fruit Fly Brain Emulation project and further projects, and I endorse it and encourage it, and I think it's something that can really benefit humanity. And like you were saying, humanity still has a lot to go, but they'll always argue and bicker until the end of time. So as well, eventually someone will have to make up their mind on what this is. So very cool stuff. And hopefully we can get more transhumanist information and debate out there. I'm going to go re-watch this and take notes on what you said that needs to be implemented into the debate context, and hopefully start integrating that into the common dialogue more and more and more. That's a really valuable resource. So thank very much, Logan, for answering that question so comprehensively. And we'll see you again next time. Thanks everybody for joining us here at Det Nation, where we're busy putting death out of business. Thank you to Gennady Stolярov. Thank you to Mr. Shoemaker, and I hope you're doing well in New Zealand, sir. And we'll have to catch up with you very soon, Mr. Shoemaker, obviously. And also thank you to Superfan Mike DeVerde keeping this show running. And of course, the stars of the show, Logan and ZION2020. ZION, we're seeing a lot more of you in the coming days. Thank you so much, everybody. And that's it.
Thanks,
Thank you.
Thank you very much.